The post-translational modification of proteins by the small protein ubiquitin is involved in many events in the cell. This study presents an original addition to the toolbox for protein ubiquitination analysis. We use enrichment techniques and mass spectrometry to uncover the deep ubiquitinome. We have made several improvements to the analysis of diglide peptides that originate from ubiquitinated proteins. These include crude peptide fractionation prior to enrichment and the application of more advanced peptide fragmentation settings in the Orbitrap. Altogether, this results in a larger coverage of the ubiquitinome. Several aspects of the protocol are difficult to describe in words. Visualization of some of the key steps is essential to be able to repeat these analyses by a different operator in a different lab. The procedure will be demonstrated by Karel Bezstarosti, who is a senior technician in my lab.
Begin by preparing cultured cells or mouse brain tissue for the experiment. If working with cells, lyse the cell pellet from one 150 cm squared culture plate in 2 milliliters of ice-cold 50 millimolar tris HCl with 0.5% sodium deoxycholate. Boil the lysate at 95 degrees Celsius for 5 minutes. Then sonicate it at 4 degrees Celsius for 10 minutes. If using in vivo mouse brain tissue, lyse it in an ice-cold buffer containing 100 millimolar tris HCl, 12 millimolar